So as a regular event sponsored by IAS, so each year our own faculty members from the campus are invited to introduce this year's Nobel Prizes to general audience on or off campus. And today we have the pleasure to have the speaker, Professor Zilong Wen from the Division of Life Science, to introduce to you this year's Nobel Prize in Physiology or Medicine. And as you know, many of you already know, this year's prize is given to James Allison and Tasuku Honjo for their discovery on the new cancer therapy based on modulation of patients' own immune system. So I think although Professor Wen's research is not directly on cancer therapy, but I think at UST, he probably is the one who knows the topic the best. He teaches immunology, and his own research is also about the immune system in zebrafish. So let's welcome Professor Wen, and I hope you enjoy the talk today.
This is my first time to give this kind of talk, and honestly speaking, that I have no idea how to do it. And in fact, I was terribly busy in the last couple of weeks for something less important. So I was just finishing my PPT like this morning. So I hope that I would give a relative comprehensive talk. But if I miss something, I apologize for that. So today, that what I'd like to do is that to celebrate this year's Nobel Prize in Physiology Medicine, for both Jim Allison and Tasuku Honjo for their discovery of cancer therapy by inhibition of negative immune regulation. Okay, so I basically, because I do not know what kind of audience I have, so basically that I divide my presentation into three parts. So I will give a brief introduction about immune system, what its systems are, and so on, and then give a short, very, very short discussion illustration that how this immuno checkpoint inhibitor therapy work. And then I possibly spend majority of time talking a little bit about, I only say that my personal point of view there in terms of and how they really come to this point. All right, so a bit introduction about immune system. So immune system basically that consists both cell and soluble factor that basically that protect us from being infected, invaded by foreign pathogen, etc. And these are the systems that are also responsible for removal of damaged cell or cell that have been turned to tumor or cancer. So I won't go through the soluble factor. I will only go through some of the key cell types that involve in the immune system. In general, there's about a dozen of different cell types that in the immune system. They are classified into two subclasses we call innate, responsible for innate immunity, or adaptive or acquired immunity. So there are different cell types for this kind to respond, and the innate immunity basically consists of few cell types like macrophage, neutrophil. These are the cells that we call more ancient, so that you find this system in much lower organism. So on the other hand, that this adaptive immunity is more advanced, that predominantly you find it in the vertebrate, but there's some lower organisms as well. Now, what I like to do today is focus on the adaptive immunity involving both two cell types. One is the B cell, another is the T cell. Now, the immunotherapy is based on the regulation of T cell. I tell you that why that makes sense and how important they are. So now these cells, basically that both innate, the cellular component, the innate and adaptive immunity, they all classify, more layman view that we call white blood cell, because they all derive from the same progenitor from the red blood cell. So the white blood cell basically are the immune system cell, where the red blood cell is the cell that is circulation, carry oxygen, and so on. All right. So now, of course, these cells that the white blood cell that they all generated in a couple of different organs we call primary lymphoid organ. Basically, they're in the thymus or bone marrow. And this mature cell, then they migrate through circulation or lymphatic system that go to basically most of the place in our body so that they can protect us. So now for this innate and adaptive immune system, there are a few differences. Okay, I'm going to go through a little bit, hopefully that you will appreciate a little bit more about the adaptive immune system research regulation, T cell and B cell. Now, the innate immunity, which consists a lot of cells, most of the cells, in fact, are white blood cells. Now, these cells, they have specificity that will recognize a pathogen. Okay, it makes sense, right? So very similarly, the adaptive immunity also have specificity, recognize pathogen, but their specificity at different level. Now, the adaptive immunity, which consists of B cell and T cell, they have incredibly highly specific interaction that recognize very, very tiny specific molecule. And this specificity, they have another very important features that they're incredibly diverse. Now, for the innate immunity, that these cells, they can only recognize very, very small classes molecule, whereas the adaptive immunity, I would say they're almost unlimited. Okay, so that's a bit crazy, but almost true. So it's unlimited. You can argue that basically the adaptive immunity have the power to recognize almost any chemical substance you can imagine, both, I guess. That's huge difference. Okay, one huge difference. The second difference is what we call memory response. Well, it was thought that the innate immunity have zero memory, but recent studies suggest that it's not true, have some kind of memory, but there's nothing compared to this adaptive immunity. So this memory is incredibly specific and powerful. So I think I would say it more laymanly to say for vaccination, that's the cell that you need to activate. Otherwise, you won't be successful with the vaccine, okay? So cell type, I won't talk about that. So key, the two different, right? So memory and then diversity and specificity. That really, I think that will be put us, in fact, that really attract a lot of scientists to spend a lot of effort to study how this system works, with the hope that perhaps by regulating this system, they allow us to treat many disease. In fact, that this regulation of this system, especially the adaptive immunity, involved in many, many of the disease you can imagine, okay? So how they work, okay? I'm going to give a few slides to illustrate how they work, okay? Now, this is the B cell, okay? One type of adaptive immunity. We have probably like 100 or 10 billion, okay, if my calculation is correct, B cell in our body. Each one of them, they have different specificity. In other words, they have receptor on their surface, very different, that recognize, supposedly they can recognize different molecule. I wouldn't say different molecule. You can recognize specific epitope, that same molecule but different region or different molecule. Okay, very highly specific. Okay, the number is incredible, right? So because each of those B cells, they have the receptor different. So therefore, when an antigen comes in, let's say a bacteria or pathogen comes in, and the first thing the B cell needs to do is to recognize it with a very, very highly specific interaction with the surface receptor we call B cell receptor. Recognize it. Specific antigen. Now, because each of them unique, then if you only have one cell, let's say you have a couple of hundred, that is not enough, right, to really gain sufficient immune response. No, you don't. So therefore, the first thing that B cell, when they become activated to do is to undergo expansion of their population. So they have to do that. Otherwise, they won't be able to produce sufficient immune response. So they expand, then they undergo differentiation into a cell we call plasma cell, which they produce antibody. I'm pretty sure every single one of you recognize this molecule, antibody. That's the key molecule that fight against many pathogens. Okay, so of course, these are terminally differentiated cells. When they done their job, they die. Well, it makes a lot of sense, right? You have proliferation, but they die. Well, if they don't undergo cell death, then we're in trouble because every single day that we activate or we come into pathogen, then we expand our population. Soon that, our body is full of this B cell, then we are gone, okay? So it's a very tightly regulated, okay? So now, T cell, on the other hand, is slightly different. So it shares very similarity to the B cell, is highly specific, highly diverse. Each of those T cells have their own receptor, recognize different molecule. In fact, I think the number of T cell is more than B cell, probably 10 times more, roughly. Okay, you can imagine an incredible cell number. Now, however, unlike B cell, which have a receptor that directly recognize the pathogen or the molecule, the T cell receptor will not be able to do that, cannot do that. So they recognize the molecule or pathogen or antigen in a very specific manner. So they have to, for some reason, okay, evolutionally, okay, if we want, we can discuss, okay, evolutionally, the receptor will only recognize the specific molecule in the context of another cell, which presented by another molecule we call MSC, okay? Don't worry about it, just a surface molecule, then with the pocket that will bind to the molecule that which T cell receptor will be able to recognize. Within this kind of interaction, now the T cell become activated, then they expand. There are two classes of T cell, whatever, CD4, CD8, whatever, they recognize different things and so on, okay? So keep this in mind. So how T cell become activated? Okay, now I'm getting a little bit deeper. Similar, right? When you activate, similar as a B cell, encounter antigen or molecule that with another cell, which we call APC, stand for antigen presenting cell. It's a specialized cell that can catch a pathogen and process in the cell and then present it on the surface in the pocket of MSC molecule. Then allow T cell to recognize it. Okay, so there are two major classes of T cell we call CD4, which have a CD4 molecule on their surface, and CD8. Now, when they recognize their specific antigen or pathogen, then they become activated, undergo proliferation, expansion, and so on. Now, they differentiate to what we call effector cell. Now, for CD8 positive cell, very straightforward. When they become activated, they try to kill the target cell. When I say to kill, for what? So let's say that you have a cell that you have a cold. Let's say you're infected by a virus. So this cell that will express their viral antigen on the surface in the pocket of this MSC molecule. Now through this kind of activation, so this effector T cell CD8 will become activated to differentiate into effector cell. Now when they look for the viral infected cell, they can find them and kill them. Okay, very straightforward, right? So that's very powerful. Now what about this guy, CD4 positive T cell? This function is slightly different. We call helper T cell. Now this T cell is in fact, this is probably the most important cell in our immune system, arguably, I guess. The reason being is that this T cell, when they become activated, they can help activation of other cells. For example, the activation of this cell need help from this guy. Not only that, I talked about earlier B cell activation, right? They produce antibody. They also need help of CD4 T cell. So therefore, you can imagine without this population T cell, you're going to lose this. You also lost the B cell response in most of the case. So that you lost acquired immunity. Then none of us can survive. In fact, that the AIDS virus, that's where they target. They target this population cell, and the AIDS patient that basically they had depletion of this population cell, they die of infection, so on. Okay, virus they evolve, very smart. Okay, so let's get to T cell. Okay, now, but this is an oversimplified model activated. So it turns, I can skip this one because T cell becomes so complicated. Okay, I can skip this one. Now the activation, when scientists, one of the key, since T cell is so important, then one of the very important fundamental question for scientists is that how T cell become activated, okay? So it turns out that it's very, very complicated. The reason being for the following. So you have a T cell receptor, which is like this one, okay? And which can recognize a molecule, an antigen or pathogen, right? In the pocket, an MSC molecule that present on the surface of antigen-presenting cell. But this interaction will never sufficient to activate T cell. No, not enough. You need second signal, we call like second signal model. You need at least one more XS3, okay? So you need what? You need another molecule which also expressed on the surface of this antigen-presenting cell, what we call B7, and another molecule we call CD28. The name is not important. It's saying that you need something else to code together in order to trigger the activation of T cell. Okay, so by doing so, now the T cell become activated. Okay, so as I told you that in adult human, that's how many, I don't know, 100 billion T cell we have. Every single day we generate about 20 million T cells. Terrible, okay? But you need to do that because all these cells, they have very short lifespan. So all the immune system cells, most of them, I wouldn't say all of them, which is not good. Most of them, majority of them, their lifespan is about even a few days until a few months. Like T cells only can survive for a few months if they don't do anything. Okay, so you need to replenish this population. Otherwise, I mean, we're in trouble, right? So I also tell you that each one of them, they're different. So they recognize different molecule, and they become activated. They undergo proliferation, activation. So the way that we control the number consistent is that they die after that. Even they activated, then they die, right? But when you think about it, only they undergo cell death is not sufficient, right? Because then you are worried about what? What about when this cell, they become extremely abnormally or hyperactive? So when they are hyperactive, they can cause disease. In fact, that's true. So when they are activity dysregulated, they cause disease because they will begin to attack our tissue. Remember, they are receptor incredibly diverse, right? So therefore, you can imagine that cell death is not sufficient to control this response. You have to be what? Incredibly balanced. So you need signal to activate, but you also have a method to turn it off in a balance. It turns out that it is true, okay? It turns out that with this kind of interaction, we call co-stimulatory activation, there also co-stimulatory negative regulation. So which I illustrate in this slide. And now that's probably the most important slide. That's the only thing you need to listen, okay? So for activation, right? So we call positive regulation. You have the receptor here. Not enough. You need CD28, beta, blah, 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 a lot of them. Okay, now the key is this guy, which is something to do with today's topic, is that you also have molecule. They play an opposite role, is that this kind of interaction, one of the molecules we call CTLA4, another one we call PD1. They also have ability to interact with the surface molecule, similar to this one. Okay, this one is similar, but this one is different, that will turn off the activation of T cell. So you can imagine that this is a balance. So therefore, it makes sense. So when the T cell is not activated, they don't express this molecule. Why? It makes sense. But once the T cell become activated, they will automatically turn on this molecule. Then you have to turn it off and so on. It's a balance. It's a great system, right? Okay, so which is true. Now, remember this, okay? Tightly regulated. Now I'm introduce the second part of talk. We call immune checkpoint inhibitor therapy. All right, so I tell you that this system is so great that you have a balance, that you always have a perfect immune response that allow us to clean or remove those pathogens but will not harmful to us, okay? But on the other hand, pathogen or on the other side, that they also evolve. So cancer, okay, of course, normally the immune system will also surveillance the cancer cell, right? If there's mutation or whatever, then they eliminate the cancer cell. But cancer cell, they evolve, okay? So that they stole this system. They basically that copied this so that you basically, when the T cell encounter the cancer cell, they turn off the T cell. Those T cells, they won't be able to activate. That's the immunotherapy, what we call immuno checkpoint therapy. That's the idea, basically. In fact, that's pretty much true. So then the argument, or it turns out that cancer cell, or mechanism is not completely known, but there's some mechanism, okay? For example, that during, well, let's say that you have tumor. So when, of course, your immune system will recognize because they are so powerful, right? So they come in and they interact with the cancer cell. But the T cell, when they become activated, they secrete a lot of factor and so on. Now, one of the factors they secrete is called interferon gamma. It turns out that the interferon gamma, so they will act on those cancer cells. So the cancer cells, they have receptor that receive the interferon gamma signal. Now, when they receive this signal, they will turn on the expression of those negative regulator molecules called like PD-1, okay? For this one is T cell. So this one will be PD-1 is on the T cell, encounter those cancer cell or other or whatever cell that they will turn on the expression of another molecule called PD-L1, which is the molecule that can interact with PD-1. Now this interaction will turn off T cell activity. So tumor have that kind of response. Or you can imagine that if we do not have that kind of response, they will be killed, eliminated by T cell. But evolution, they do that. Only those that have this kind of ability, they survive. Okay, you can imagine, very simple, right? So then they survive. So with this kind of discovery, then the idea, so the immunotherapy, that which they give Nobel Prize to, is that very simple, right? With this kind of discovery, then the idea is that if you block this interaction, you reactivate this T cell, so they allow this T cell to eliminate the cancer cell. The idea becomes very simple. That's it. So it turns out, basically, that's what they did, okay? So the idea, what we call immuno checkpoint therapy, which is the reason they gave Nobel Prize to this year, it worked as the following. So what they did is that they generate molecule, in this case, it's an antibody, okay, that will recognize, one of them will be recognize this CTL4, okay? So with this, it will block this CTL4 interaction with, let's say, that another molecule called B7. Most of the case there, in either cancer cell, in most of the case, actually in this case, it will be an antigen presenting cell, which is required for activation of T cell, okay? So similarly, they also develop antibody that will react with this called PD-1 that will block the interaction between the T cell and the cancer cell between them, because the cancer cell, they will turn on the expression of PD-L1 that will interact with PD-1 that will shut off T cell. But if you block this interaction, you prevent this. So you allow T cell to become activated, you eliminate the cancer cell. Okay. In fact, that these two antibody, anti-CTL4 and anti-PD-1 therapy, they have different mechanism. Even though they sort of reactivate the T cell, but they activate different pool of T cell. So I remember, right, there are CD4 and CD8. Now the PD-1 antibody predominantly will activate the CD8 cell that have been infiltrated in the cancer environment. But under normal condition, because the cancer cell, they express this high expression level, this molecule, this T cell, they put into sleep. They don't respond, and they don't die either, okay? So, but with this kind of blocking, then you reactivate those T cells, then you kill, okay? On the other hand, the anti-CTLA4, this antibody works slightly differently, because you can imagine that in this case, it will block this kind of interaction, not the T cell directly interact with the cancer cell, rather directly interact with this kind of presentation, which will lead to activation of CD4 T cell, which is also critical for activation of CD8. So, for example, if you combine these two therapies, presumably or depend on individual patient and the situation, okay, you probably have synergistic effect, okay? But I haven't seen the data or any publication for the combined therapy, but right now, majority of them, they are single therapy for either one of them. And some of the patient is 100% recover, but I would say that the response is only like 25% for some of the cancer and so on. Okay, it's not a perfect way, but you can imagine that will be one of the incredible or fundamental principle allow us to put forward that the regulation of this kind of immune response look to be very promising for the future or develop a new drug for treatment of cancer, perhaps other disease as well. Okay, so that's pretty much for, I guess that's pretty much just all you need to know about this therapy and so on, because I think the knowledge is basically that's it. Okay, you know the immune system a little bit, you know how it works and so on. Okay, so now for the rest of the talk, if you already have enough, then you can lay back and sleep, okay, or take a nap. So now, because the rest of the part that I will talk a little bit about how they get here, how do we get here, or how did they get eventually to discovery and get a Nobel Prize, okay? So it's my historical view. I mean, I may miss something, but hopefully that I get it correctly. Okay, I'm going to go through a little bit, okay? So, especially I think for students, okay, I guess the take-home message will be that, okay, and if you want to be very successful, I mean like hardworking, whatever, everyone work hard, right? So you need a little bit more, okay? You need, I'm going to say that you need to be intelligent, whatever. I think everyone is very, very intelligent. Okay, you need a little bit extra what? You need to have the guts to go for big question. Okay, that's one thing. You have to do that. Otherwise, it will be very difficult. You don't even have the chance. Okay, so it's like, how do I know the big question? Of course, sometimes you don't know, but sometimes it becomes very obvious. Okay, go for big question. Second, I don't know, I cannot help. Sometimes you need luck. You need to be quite lucky, I guess. So hopefully that by the end of talk, you'll appreciate a little bit, okay? And of course, the last take-home message I want to send is that, okay, in order to all deal with all this disease, I think fundamental basic research become very critical, if it's not more important. So because you will see that on the way that I illustrated, without all this basic discovery, none of this will happen, okay? So let me go through a little bit historical point of view called immune therapy. I mean, in fact, that we discover, or scientists have discovered, that somehow immune cell that may involve in sort of like cancer surveillance. Okay, the reason being is that about more than 100 years ago, okay, that and scientists have observed that in the solid tumor, there's an infiltration of immune cell. I mean, immune cell. I mean, a cell that looks like immune cell back then, right? It's not very well characterized, but you can trace back that there's some kind of indication. So, of course, with the advance of science, you see that there are a few, turning points that which give us great hope, but it doesn't really work out, actually. I think one of the first turning points, one of the first turning points is the discovery of cytokine. Okay. For example, this one. Cytokine basically are the secreted protein that will regulate the immune system. Okay, majority of them, they play a positive role, but they also negative role. So it was thought that cytokine will be a great, because you can synthesize this protein easily. You can also administrate it. Okay, you can sort like this. Okay, it was thought that because in fact that in the 90s, okay, interleukin-2, one of the key factors that regulate T cell activation, has been used for treatment, at least for clinical trial, for many cancer. Okay, I mean, it's just really big hope, but but it turns out very disappointing, okay? And in fact, majority of those trials fail, okay? I think the reason being is that, well, we just do not understand the immune system enough, okay? So I think that's one of the big holes. I think the second, I think very promising, I'm not sure if it is here, is that it's the development of monoclonal antibody, which is very useful because whatever I talk about to block those interactions, they are monoclonal antibody. What do I mean monoclonal antibody? Means that I told you that each of those B cells, right, they have different receptor. So the receptor and antibody are encoded in the same gene. In other words, that if you have this kind of receptor, you will produce the same antibody that recognize the same molecule. So because each of those B cells that have different receptor means that they produce different antibody. They're unique. So antibody produced from one single B cell we call monoclonal antibody because they're so specific, right? So the development of monoclonal antibody, I think, is 1975. So of course, people that developed the technology received Nobel Prize for medicine as well. It was also give a big hope because I think, very dominant view that, now finally we have a target, right? So let's say that if we can find a tumor-specific antigen, so we can develop monoclonal antibody, we kill the cell, right? I mean, very simple. In fact, very disappointing as well. In fact, when I first went to USA, that's what I was doing. So I spent a couple of years working in the lab, trying to develop monoclonal antibody against some kind of tumor antigen. Of course we fail, okay? Not only we fail, many labs have failed. Very disappointing, okay? So, but of course, it took us a long way. It's not like it's not useful, very useful, right? But it took us a long way to get here. So now, I'm going to talk each one of them, Allison and Honjo, okay? It's because their work that led to the immunotherapy, which is, I think, have huge impact. So I'm going to do very specifically to talk a little bit each one's work and how, their career a little bit. I hope I try to dig out as much information as possible. So, okay, I'm going to talk a little bit first about Tasuku Honjo first, okay. So Honjo is, he's always, not very much to talk because I think he's always first-class student from, primary school. I mean, for his first grade or second grade, he already finished like 4 or 5 grades, everything. I mean, he's, I guess, not very much to talk about, okay. And he was so good that, so then he was admitted to, the medical school and graduated with a medical degree. So I couldn't find the reason that why he decided to pursue PhD. I tried, okay. But somehow, and then he decided to pursue a PhD that to do biomedical research, okay, in Kyoto University. Now, in fact, and he was so outstanding that he received scholarship when he was a graduate student that allowed him to really study in the USA. In fact, in Donald Brown's lab, one of the leading scientists, okay, incredible, study, ribosome RNA. I mean, so where that he basically learned a lot. So it's very interesting, right? So then after that, he did a short postdoc in Philip Leder's laboratory, who also very incredible, famous. Okay, he should. I wouldn't say should. He almost win Nobel Prize. Okay. So a couple of years. So and then began to study immunity. So very funny. In 1974, he became assistant professor at University Tokyo, and then but he only received his PhD degree 1975. I tried to find out the reason, but I couldn't. Okay, I don't know why. Perhaps they said need publication like us. Okay, yeah. So, but of course he managed to publish a few publications in Philip Leder's lab. Very nice work. Okay, and after that, within a few years, okay, he had made some incredible contribution in the field of immunology. And then soon he moved to Osaka University, become full professor at the, well, I say the age of 37. In Japan, I don't know, we have faculty in Japan, right? They are incredibly, I mean, they are terrible hierarchy. So you want to become a full professor, it's almost impossible unless you are very senior. But by the age of 37, he was a full professor. I mean, I think that's very, truly an honor. So then he returned 84 to Kyoto University, where he graduated, and then continued his research. In the Kyoto University, he discovered the PD-1 and PD-1 ligand, or L1. So and then the interaction that involved in tumor genesis. So I'm going to go through a little bit his achievement and then go into how he make it. Okay. So before I do that, again, I have to go through some of the background. I think it's intriguing. Okay. So I cannot stop myself to mention a little bit. Okay. So Carl Stanner is I think one of the greatest immunology, probably in the last century, I think. And he's the person who really provide evidence to show that our immune system, or acquired immunity, or B cell, have incredible diversity. In other words, that he have designed experiment to prove that the B cell in our body or animal are capable to recognize almost any chemical substance. So I think it's incredible. He basically laid out the key foundation for whatever later on we have done. Okay. So of course he received a Nobel Prize. Not for this, he's also responsible for discovery of human blood group. Of course, when he received a Nobel Prize, he basically said that he would rather the committee to give Nobel Prize to his discovery of antibody diversity. Okay, with this in mind, okay, then allow us to have an idea to generate what? Specific antibody or monoclonal antibody recognize specific molecule. Otherwise, none of those are achieved. Okay, so a little bit more history. Okay, so when you think about that, right? Once the antibody diversity have been illustrated, you can imagine. I mean, all the scientists will try to jump into to understand antibody, because that's become so important, so promising for, let's say, treatment of many diseases, because you have a lot of imagination, right? So therefore, the structure of antibody become very attractive for young scientists, particularly for young scientists. So, and there are a couple of them, Edelman and Porter, and they are the ones that to use very simple enzymatic digestion to basically figure out the antibody structure, okay? So which consists two polypeptide chain, light chain, which is this one, and then a heavy chain, a larger one. So each of antibody have two of light chain and two heavy chain. They also know that by using enzymatic digestion and antigen interaction, they also shown that and each of those antibody that have two functional regions, one we call constant region, which is 2/3 of this molecule. Now, the reason we call constant region is that even though each of those B cells, they produce different antibody, which recognize different molecule, but this constant region, 2/3 of the molecule, they're identical. Now, the only difference is the region we call variable region, which is right here. Okay, that's the region that will contact the molecule or the antigen. Okay, so very important discovery, right? Because then you define a lot of things. So now, the key question is that how diversity in antibody is achieved. So a lot of, scientists try to chase this question, same as Honjo. When he went to Philip Leder's laboratory, and he said that, wow, that will be the one of the mystery that try to say, well, perhaps we should try to understand how this diversity can be generated, even without knowing very much about our genome. We know that our genome only have 25,000 genes, right? But antibody, right? I tell you what, we have, 10 million of it, right? So it's incredible mechanism. So he tried, actually. He did publish one of them, a couple papers. I'm going to list one of them. So he basically did, I won't go into detail. I must confess, I have not really read in the detail, okay? Even you ask me, I'm not 100% sure I really know. But I think what he did is that to look at the light chain, the smaller chain, look at the organization of those chains in the genome, which is not simple, not trivial, very difficult to do back in 1974, right? But he was able to do some experiment to really show, let's say, the copy number and perhaps a little bit their arrangement in the genome. Okay, of course, I think he managed to publish 2 papers in PNAS, which is very, very good, okay? So of course, the ultimate goal is to figure out what's the underlying mechanism to generate such a diversity, right? So the whole lab, field leader, they're trying to do that, as many other labs as well. But somebody else ahead of them. So as a Japanese guy called Susumu Tanigawa, in 1976, he figured out the mechanism. So I won't go through the mechanism, but just say he did that. Of course, he received Nobel Prize. So basically, the competition is tough, right? But it's not the end. It's not the end of the world, right? So I won't go through the mechanism. So then he decided to move back to Japan after that. So I think in 1974. So I read a little bit his biography, which is in Japanese, and then I have to put it translate into English, and the translation was so bad I can read a little bit. So I understand a little bit, but basically I try to understand that how he started his own lab. It turns out that when he started, it's incredibly difficult. I think just like 30 years ago, if you went back to China, something like that. You had no money, no whatsoever, and then you're assistant professor in the hierarchy of Japan. Basically, you're under a professor, basically. So, but I think he's talented enough and persistent enough and also lucky enough that he really make it work. So the first key discovery for him, again, you ask big question, right? So the first big question is gone, right? Because somebody else already figured out the mechanism, diversity is gone. So what else he wants to do, right? He basically said that, well, other than diversity, he realized that, well, there's still different subtype of antibody we call IgG, IgM, IgA, blah, blah, 5 subtypes. Now these subtypes are determined by choosing a different constant region. So whatever I tell you previously is not completely true. The constant region have 5, okay? Actually, it'll be more than 5, but that's all, okay? But the rest will be the variable region. So, and he also realized that there are very well-known study that for immune response, so we have 2 different types, well, 2 different phases. We call primary response and secondary response. What does it mean? Primary response means that when the host encounter a pathogen on the first time, so then you have immune response, right? You produce antibody, right? Let's look at antibody. So under this condition, you generate predominantly IgM. That's one type of antibody. It makes sense because the B-cell receptor is IgM, okay? Which have membrane bonding. So the response normally are weak. So and then if you let's say that you recover, 1 month later or 2 months later, then you'll be infected by the same identical pathogen. Now, because you have memory, I told, right? The immune cell that become activated, some of them, they will not die. They transform. They convert themselves into what we call memory T cell or memory B cell. And some of them can survive, get more immortalized. They can survive forever. That's how we can, lifetime vaccination. That's how it works, okay? So, but second time, now these memory T cell or B cell, they very quickly they respond. And normally they generate much, much more amount of antibody, but also in most cases, they won't produce IgM anymore. They switch to IgG. That's we call class switching. Oh, okay. Honjo said that, well, nobody knows what is going on. Maybe I will do this. So he did that. Okay, in fact, So I won't go through detail. He basically single-handed, he basically identified the mechanism. So basically simple experiment. If you look at nowadays, very simple, but back in 1970, that's not that trivial. So he was able to show that the class switching is due to a mechanism or deletion of those DNA. Let's say that you have different constant region called heavy chain, right? Blah, 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 different type. And then the first one is this one, IgM. That's the primary response. But during secondary response, this region will be removed. So therefore, you express the second one, so on. Okay, that's how he select a simple mechanism. So not only that he figured out the mechanism, he also figured out and what will be the sequence and what will be the enzyme that mediate all these things. In other words, that he basically single-handed to basically elucidate the entire mechanism or class switching. And he identified the enzyme, of course, AID. Okay, that's the enzyme that responsible for this class switching. They are also very responsible for another very important, we call somatic hypermutation. I won't talk about. And he also will be the person that identify interleukin-4, interleukin-5, two of the cytokines important for this pathway, and then interleukin-2, one of the interleukin-2 receptor. Okay, these are all very important discovery. So in very short period of time, he became very famous. Okay, no question about that. Okay, so his contribution enormous. Now, what about? But these are not the discovery that led him to win the Nobel Prize. It's the second one. So the second one, which is the identification of this molecule called PD-1, stands for Programmed Cell Death Protein 1. Nothing to do with the function, right? So you can imagine that initially he had no intention to, let's say, to study a molecule that can involve in cancer or whatever. No. All he wanted to do is try to understand the regulation of T cell. Now, what kind of phenomena he wanted to understand? That it turns out that there are few T cell lines. Those are all tumor cells. Many of those tumor cells, they are not fully mature T cells, but they respond. There are a couple of them he chose. Okay, it doesn't matter each one of them. Now, these T cells have a very interesting property. So, for example, this T cell 2B4, if you stimulate with a common antigen we call PMA, don't worry about it, it's a polysaccharide protein. Polysaccharide, you can activate T cell, many B cell as well. Okay, so in contrast to those normal T cells, when you treat this PMA to T cell, they become activated, they undergo proliferation, right? That makes sense. But this guy, instead of undergo proliferation, they die. They commit suicide, what we call a programmed cell death or apoptosis. It's a kind of cell death that will commit suicide, okay, it's like this, very specialized cell death. Similarly, this is another T cell line, have similar response, similar phenomenon, but different response. These cell lines that normally, in order to grow them, you have to put a growth factor we call IL-3 to maintain their survival. But if you take off this one, they also undergo this kind of programmed cell death. So now all they want to do is that, they try to understand why this cell, when you give PMA or you remove interleukin IL-3, they die. That's it. That's all they want to do. So they took a strategy we call differentiated screening. I won't go through detail, but they are trying to identify molecules that involve this process. It turns out that they identify one molecule, which is this one. That's why they call programmed cell death protein 1. And what this protein, or what this gene have shown is that if you treat this cell with PMA, their expression will be what? Incredibly enriched, okay? So the expression, in other words, that the interpretation is that expression, this guy will trigger the cell death of T cell. Okay, this is similarly, same thing. That's the conclusion they have. Their paper published in EMBO journal in 1992. But this is nothing to do with negative regulation of T cell yet, okay, at this point. So it took them a few years to figure out what's the function of the true function of this gene by using a very, very straightforward technology. I think every single one of them you can imagine, you can also use the same technology or same approach as well. Is basically that they deleted this gene in the mice and asked what happened. So they generated, which is probably 99. Okay, that's 7 years later. Well, genetic modification animal was in the 90s, not that trivial, okay, pretty difficult. Normally it takes 5 years for a student to do. It's not surprised take them 7 years. So what happened is that in this the PD-1 deficient animal is that they develop autoimmune disease. And they further show that the reason this animal have autoimmune disease is not they generate more T cell. No, they have similar number of T cell, but rather that this T cell, the activity much, much higher. Now this will give the first hint that the true function of PD-1, they propose that this molecule, which expressed on the surface of T cell, that act as a negative regulator of T cell activation. Now that's the first hint. And I mean, that will give them, I think, the first important clue that what this molecule does. But that's not sufficient, right? So, I mean, obviously, if you are the one that work in this project, you probably want to know why. I mean, because if this is a membrane-bounded protein, you would definitely think that, well, this molecule must bind to something, right, in order to suppress this T cell. Otherwise, they'll become activated, okay? So the hypothesis is that they put forward a hypothesis that there has to be another surface molecule that on the surface of antigen-producing cell. That's their hypothesis. Because remember, T cell activation, they need receptor that recognize the antigen or the molecule that presented by an antigen-presenting cell. That's their argument. So later on, they went on to identify that surface molecule we call PD-1L or PD-1 ligand. It's called PD-1L in collaboration with Gordon Freeman at Harvard. Okay, so 2000 actually, they published this paper in 2000 in Journal of Experimental Medicine, and they showed that true. If we inhibit this interaction, the PD-1 interact with PDL, if you block this interaction, you basically you perturb the activation of T cell. You can look at their proliferation, you can look at the growth factor they produce, and so on. So in other words, so if you block this interaction, you trigger activation of T cell. If you activate this kind of molecule, then you suppress. Very, very convincing data. That's 2000. Okay, I'm trying to emphasize the time again. Okay, then 2 years later, because by then they basically have the idea that perhaps, okay, this kind of interaction could be utilized in tumorigenesis as well. The reason I think they have the idea is because Jim Allison's work. Because in '96, Jim Allison already showed that CDL4 with the ligand interaction B7 will link to tumorigenesis and so on. So it's not surprised they do that. So it turns out that if they use antibody, which is block, antibody recognize PDL1. Now this interaction, okay, you can block this PDL1 interact with PD1, okay, this interaction. With this, you can block tumor growth in animal. And so I won't go through detail. That's it. That's their demonstration. With this in mind, basically, they set up this kind of, ideas that, well, you can utilize this strategy, which is an monoclonal antibody, which recognizes very specifically this molecule. You can block this negative regulatory pathway, then you can activate, presumably T cell, then you can eliminate the tumor growth. So that's the idea, okay? So with this, it's pretty much complete for them, right? But I want to talk 2 more slides. Okay, now I told you, right? They identify the PDL1 2000. But I can tell you somebody else ahead of them. Okay, Chen Leping, okay, at Yale University. He's also very well-known immunologist. He also has similar finding. In 1999, his lab have identified a molecule which he called it B7H1, which is identical to PDL1. It's the molecule that binds to PD1. They published this paper in Nature Medicine. Well, 1 year ahead, okay. But I must say that at this time, at this paper, they did not know that whether this H they call B7-H7, whether this molecule bound to PD1 or not. They did not know, okay. But they identified this molecule, and they showed that they are important for regulating the immune response. But they did not show their interaction, okay. So, but the 2000, right, they are Honjo's group, they show their interaction, okay, later, okay. But in 2002, okay, Chen Leping's group also showed that, of course, he realized that. He also showed that this molecule is PD1L interact with PD1. They play a very important role in the surveillance or immune system for cancer. In fact, he proposed the model that the expression of PDL1, because he found that many of those cancer cells express high level of PDL1. He proposed the model that by expression of PDL1 in cancer cell, they allow this cancer cell to escape the immune system. In fact, that they published this paper, and I think 1 month ahead of Honjo. So it's your judge that whether he should receive a Nobel Prize or not. But I think that his contribution cannot be denied. Okay, so let me turn to the last part of talk. Jim Allison's contribution. Okay, he's the one who really, identified the other pathway. Okay, which is CTL4. Okay. I think Jim Allison is worth to talk a little bit, okay, because he have interesting personality, I think. So based on his biography, okay. So apparently he's a very talented, very smart boy even when he was very young, but he has such a strong personality that he was interested in biology because his father is a doctor. And then in high school, and he refused to take biology, at least he refused to attend biology class because the reason that the teacher will not talk about evolution. He said that I'm not going to attend these classes. Forget about that. So because this, right? The school almost want to kick him away, okay? If it's not his father, he'll be already dismissed, okay? So what happened is that then they make agreement that allow him self-study biology in a sport hall. Self-study, okay, of course, occasionally there are, senior students that try to teach him a little bit. That's how he learned biology. But I guess, he's pretty damn good, I guess. At the age of 16, he went to college. Well, he grew up in Texas, so University of Texas in Austin, okay? So in a program we call pre-map. So pre-map is a highly elite program. So you have to be very, very top student in order to enroll in a pre-med student, okay? But very soon that he decided not to continue, even though his father is a medical doctor, he would prefer to switch to science. That's what he said, I quote it. So I realized that, going the pre-med rule means that memorize a lot of information. Okay. Oh, come on, everybody have to memorize a lot of information. And as a doctor, I think that may be the key point, I guess. You cannot make mistake, I guess. You cannot make huge mistake, I guess. And you have to know what to do. But on the other hand, as a scientist, you are supposed to make mistake, yeah, I guess. True. And hypothesize, and then test it and so on. So whatever he said, okay. And last thing that, plus my grade in organic chemistry suck, okay. What means that he's not like, he's not like good at everything. Well, so I think his personality even illustrated, further I think illustrated as when he went to graduate school. So he was accepted in the same university graduate school, okay, University of Texas, okay. But there are a lot of debate whether they should accept him to the PhD program because, he had a lot of classes that they fail. The reason is strong personality that if he doesn't like it, he never bother to attend, and he never bother to drop. So then you get 0 point. So either they have A or you have F or fail. But I think if he's in China, I think he's not going to make it, I guess. I don't know, but I think it's interesting. Okay, very strong personality. So after graduated, so he went to Scripps, okay. So Scripps Clinic and Research Foundation in San Diego for his postdoc, which study biochemistry, actually immunology. But his only focus is to study, you see, I told you the T cell, why? They only recognize the antigen that bound in the surface of another molecule presented by other cell called MSC. So the MSC that they bound to small molecule, in fact, they're peptide. So he was studying this kind of interaction. So how the antigen bound to MSC molecule using pretty classic biochemical approach. So it's 1977, so he assumed a position, assistant professor at Texas, okay, Andy Anderson, okay, where one of the largest cancer center in USA. So junior faculty, okay, so according to him, okay, so his biography, in the beginning, he had no idea what to do. In fact, I read his publication. Actually, he published one Nature paper about those MSC and antigen interaction. Pretty good, I guess. But apparently that he wasn't that satisfied. So I guess he does not really want to follow his previous postdoc supervisor's rule. He want to do something new. So he was trying to say, oh, maybe I should do something else. So I tried to figure out what is important. So suddenly, if you go back to the history, right, B cell was very important, but diversity had been, gone. He said, well, B cell, then nothing is important. He said, well, I'm going to work on T cell because nothing know about T cell receptor. He said, that's what he wants to do. See, big question. I'm pretty sure that's the case. So of course, he had no idea how to do it. So I read his biography. He said that one time he went to a seminar which is delivered by Irvin Weissman, my former supervisor, okay, personal supervisor. And after he lectured, he realized how to do it. So when I read how he, do the T cell, I realized, yeah, maybe that's the way. So what he decided to do is the following. He decided to say, well, I'm going to work on T cell, but because I know nothing about T cell, what I would like to do, he said that, well, I need to have a handle. So the handle is beneficial by this. Okay. Two scientists, I told them, in 1975, they developed a strategy to generate what we call monoclonal antibody. Means that you can use, I won't go through detail, you can use this kind of strategy that you can generate unlimited source of monoclonal antibody from one single B cell. Okay. So I won't go through detail how they did it, but that's it. So he said that, well, maybe I can use this same strategy, not try to kill cancer, but try to identify the T cell receptor. So what he did is that he basically identified or developed monoclonal antibody that will only recognize T cell, but not other cell. Means that he have chance to recognize a T cell specific molecule that could be T cell receptor, that could not be, right? But that's the way he did that. So it turns out that And he think that this antibody recognize T cell receptor for what? Without sequencing, well, you have no idea, right? So the key is the following. He did a lot of experiments. He showed that T cell receptor consists 2 polypeptides, which turned out to be true. This one, you have alpha and beta chain. These 2 polypeptides are held by disulfide bond. He has shown that experiment. I won't go through detail. Very, very good. But what does he know that without sequencing, how do you know it's a T cell receptor? Well, that relies on prediction. So the prediction is that, it will be that T cell receptor should be similar to, the T cell receptor should have a very similar structure as a B cell receptor for the following 2 properties. The first property is diversity, means that every single T cell, their receptor are different. Okay, the variable region. But they also have constant region. They also share identical sequence. That's the key. So if you buy this hypothesis, he will tell you that whatever he have seen these 2 polypeptides are T cell receptor, for the reason being that he used enzyme, trypsin, to digest this protein, each one of them. Then he ran the 2-dimensional gel and to compare the T cell receptor derived from different T cell. So all of them, they are cancer cell, but they have T cell receptor. So the assumption is that if the hypothesis is true, you're going to see that even though they derive from different T cell receptor, they should share, many of them, they share identical peptide. In other words, their pattern are identical, means that their digestion, well, I think it's pretty cool actually. So it turns out true. And there are all these black, they are identical. And there are also some different. So there's some of the peptide, they are different between different T cell receptor, or whatever. Based on this, he made the conclusion that T cell receptor consists 2 polypeptide chains, alpha and beta. One is about 45 kilodalton, the other one 35. And they hold by disulfide bond, blah, blah, blah. It turned out everything is correct. Of course, I mean, the next stage, if you are the one who do this, is that what you want to do? You want to clone the gene, right? Of course. He wanted to do so badly, but he was beaten by someone else. Okay, I don't want competition again. Yeah, someone else cloned the T cell receptor. So one is Tang Mak, well, it's grown up from Hong Kong, actually. And then Mark David. I won't go through it. They are the one who cloned the T cell receptor. So, I mean, I think sometimes it happens, right? So, but if you are scoop, okay, it's not the end of the world, right? But I think I can predict if Jim Allison, if he's the one that he identified the T cell receptor, he will continue to work on T cell receptor. He probably not going to win Nobel Prize unless T cell receptor they going to get Nobel Prize. See, okay, it's not the end of the world. So now turn to the CTLA-4 story. Okay, initially CTLA-4 was identified long time ago, 87, by somebody else, not by Jim Allison. It's by a French scientist, okay, Pierre Goldstein. Okay, he's also a very well-known scientist. Okay, and he cloned this. It's based on homology, have certain structure we call immunoglobulin superfamily. Means that there are thousands of proteins have this kind of structure. Okay, but for some reason they clone this. Okay, and they clone this. They publish in Nature without knowing what this molecule does, but they know that this molecule expressed on T cell. And they know that only when the T cell become activated, this guy is on. But they did not continue to pursue this molecule. Otherwise, maybe the story will be fitting or wrong, okay? So who will be the first one to demonstrate the function of CTLA-4? Not Jim Allison. Somebody else, again, see? Called Jeffrey Bluestone. I think then he was at University of Chicago. His laboratory was the first one to demonstrate that CTLA-4 as a negative regulator for T cell activation. Similar strategy. He basically generate antibody that recognize T cell. Turns out that have molecule that recognize this molecule. So by using this antibody, he was able to show that, and if you activate this pathway, I say activate, so you can, when you use antibody, there are two consequences. When an antibody binds to a molecule, a surface molecule, could lead to activation of this molecule. Depending on how you design. You can also lead to block, whatever you can design. You can easily design this kind of tool, okay? So in this case that he designed a molecule, or antibody molecule that you can activate it. When you bind to this molecule, it activates this molecule. It will inhibit the T cell activation. So his lab basically provides the first evidence that CTLA4, which is a membrane-bounded protein, expressed on the surface T cell, that act as a negative regulator. They also show that this molecule will only express when T cell become activated. So everything makes sense. Like you activate it, that's the one that acts as a negative regulator to whatever. All right, so here come to the lot, okay. So Jeffrey, I mean, he makes such an important discovery, right? But he was not interested in cancer. So he was more interested in like autoimmune disease intolerance. So he basically studied the role of CTLA4 in immunotolerance, which also makes sense. See, I mean, sometimes luck really contribute a lot, okay? But on the other hand, Jim Allison, that he also studied this molecule, CTLA4, and I think independently, 95, okay, I read that his paper was submitted in 94, but for some reason, it took him 1 year to publish. I don't know why, because it's very common right now. I mean, for us, it took 1 year to publish, but in the 90, 3 months, or 6 months is very long already, okay. 1 year, that's pretty long. But nonetheless, okay, Jim Allison, I think I should say that independently, also uncover the function of CTLA4 as a negative regulator. I mean, he also showed that this molecule will only express when T cell become activated. I don't have to go through detail. Okay, so they showed that if you activate this molecule using antibody, when we say cross-link antibody, it's a cross-linking antibody that you can cross this surface molecule, they dimerize them, you activate this pathway. So then you inhibit the T cell activation by looking at their proliferation and so on. And he further showed that and CD4 will compete with CD28. Remember, that's another molecule that has a positive when it bound to B7. So in fact, it turns out that CTLA4 also bound to the B7. They compete with the same like ligand. So depend on which one. So if this one is being expressed, then you compete out. So you turn off the T cell. So I guess he also contributed, okay. But he suddenly turned his attention to cancer. Okay, initially, all he wanted to do is to understand how T cell activation become regulated. I don't think he had intention that he worked on cancer. But with this kind of, knowledge, in hand, that he turned his research interesting to cancer. Partially, I think because, according to his biography, his mom was dying cancer when he was very young. His uncle as well. So I guess that really triggered him to turn his research attention to cancer. So 1 year later, 96, he basically published. I think that's the most important paper, okay, for his contribution. Published in Science. He basically showed, 96, he basically showed that he developed the anti-CTLA4 antibody. In this case, it's not the cross-linking antibody. Okay, cross-linking antibody is not good, that will be activated. This is a blocking antibody. So when you bind to the CD4 molecule, we will block the CD4 bond with other molecule, okay? Therefore, block this pathway. So he have shown that if you inject this antibody, either before or together or after you give immunization of tumor in the animal, you can reduce the tumor size or sometimes you completely eliminate the tumorigenesis in animal. So this is astonishing, okay? With this, he immediately proposed a model. I think sometimes it's also a lesson. When you have something novel discovery, please propose a model. Well, even if it's wrong, it's okay. But if you're right, then you're going to what, right? So he proposed a model called immune checkpoint. Okay, say that, well, during T-cell activation, all this that has to be checked, we call like immune checkpoint, just like cell cycle. You check whether you should go to activation or rest. So these molecules, CTLA4, is one of the molecules that act as a brake to check whether the T cell should undergo activation or remain as inactive. He proposed model. Of course, he put forward the possibilities that, well, if you can regulate this checkpoint, you may be able to eliminate cancer. Of course, later on he proposed a model called checkpoint inhibitor drug. So as simple as that, basically that using monoclonal antibody that block the CTLA4 interact with B7 molecule. By doing so, you can eliminate the tumor. Okay, in fact that, well, it worked, okay. Well, not every single cancer, it really depends on individual. So the rest will be history, right? So, and the first clinical trial, I think is 2000. This is the first anti-CTLA4 antibody, monoclonal antibody, first one. And then it took them 10 years, okay. So 2011, FDA approved the first successful or first drug of anti-CTLA4 antibody to treat melanoma. But if you look at them right now, there are many of those antibodies. Majority of them, they are the antibodies that recognize PD-1 or PD-L1, or because they are the ones that directly interact with cancer cell and so on. So by doing so that, I think it's become one of the very promising strategy to treatment of many cancer. So I think I'm looking forward that there will be more similar, I'm not saying that there are antibody, but similar drug that develop for immunotherapy and like CAR-T, whatever. Well, CAR-T, also one have been approved. So by doing so, that I hope that today that I convince you that basic research, fundamental research, in order to treat human disease, you need to understand basic science. And plus, you also need to have the guts to believe what you think. So pursue translation as well. So thank you very much. Okay.
Thanks. So I have a very nice and inspirational talk. So I have two questions. One is academic and another is non-academic. So academic first. So I was wondering why antibody of CTLA and PD-1, injecting them will lead to the hyperactive immune system, right? So I was wondering why that will not cause some, autoimmune system, some side effect apart from suppress the tumor. First question.
Yeah. Well, that's an interesting question. Yeah, I think, of course, there are definitely side effects. There are two potential side effects. The first potential side effect is that antibody is a protein. So when you inject, that counts as a foreign antigen to the host. So the host will generate immune response. So I wouldn't say that will be definitely a side effect, but at least that will basically diminish the effect of the drug. Now the second potential side effect is, like you said, that there's potential possibility that the immune system that is dysregulated, they're hyperactive. So that may lead to some, but I guess you probably gain more, right? When you have cancer, right? I guess, autoimmune disease, then you put this aside. But definitely there are potential. Yeah, but also the drug. I think the antibody you give. You can control, right, the concentration. You can monitor. I think it's not going to be a huge problem as well. Yeah.
Okay. So second question is non-academic. So just very impressive about the discovery history of this to outstanding scientists and about how they identify the important big question in the field. So I was wondering, for a student like me and other student, do you have some suggestion about how to identify the big question in the field? I mean, I'm not from Nobel Prize, but just a lot of scientists suggest you should focus on the big question and do it. Yeah.
I guess it's easy. Well, I must confess, it's easy to say, right? It's difficult to do. Okay, so I think what we call it mahopa. Okay. I guess the only suggestion I have is that I think don't be afraid to take risky project. I think that's when you are. I think even when in your career, I think, yeah, as long as you can do that, I'm pretty sure that you have chance that you do well, okay? Well, I say that you have chance, right? Doesn't mean that you'll be successful, right? I think they depend on a lot of things. Also luck as well.
Yeah, it's a very nice talk, Zilong. So I just, out of curiosity, you just put this in a context of comparison with a small molecular compound cancer drugs, right?
Yeah.
When 20 years ago, when the first medicine, Gleevec, was on market, everybody's so excited. But later on, this tyrosine kinase inhibitor, they have issue with the drug resistance, developed quickly after that. Do you see the immunotherapy also have the antibody resistance that could be developed?
Well, that's also another very outstanding question. So, well, you may imagine that's still possible. Yeah, you may imagine that's still possible. But perhaps we may be able to get around because you're activating the T cell rather you act on the cancer cell itself. But of course, maybe the T cell can be tolerant to the molecule as well.
You think it's on chemistry point of view, this interaction surface is much larger between the antibody and
but because you're not really directly targeting the cancer cell, the molecule itself. So perhaps you're better off, but I'm not 100% sure. Okay, but I think it's too early to really make the argument because I think you need some time to really look for maybe another 5 or 10 years, when you accumulate sufficient data, you probably know, I wouldn't surprise, you may also have to deal with this kind of problem as well, If you know, okay, thank you very much, okay.